Today's video we will talk about the famous gene editing technique which is CRISPR-Cas9. This technique has ability to change the organism's DNA. In this technique, genetic material can be added, removed or altered at a particular location in the genome of plants, animals and even humans. Before the discovery of CRISPR-Cas9, scientists relied on two gene editing techniques, that is zinc finger nucleases (ZFN) and transcription activator like effector nucleases or TALEN. ZFN has a zinc finger DNA binding domain which is used to bind a specific target DNA sequence. It also has a restriction endonuclease domain that is used to cleave DNA at the target site. TALEN also composed of DNA binding domain and restriction domain like ZFN, but their DNA binding domain has more potential target sequences than ZFN. Both techniques are expensive and time consuming for researchers and manufacturers. And then there comes the CRISPR. So what exactly is CRISPR? CRISPR stands for clustered regularly interspaced short palindromic repeats and refers to unique organization of short, partially repeated DNA sequences found in genome of prokaryotes. CRISPR and its associated protein, which is Cas9, is a method of adaptive immunity in prokaryotes to defend themselves against the viruses or bacteriophages. So before discussing the mechanism of CRISPR, let's see how the system works in the adaptive immunity for bacteria. During the adaptation process, bacterial cells become immunized by the insertion of short fragments of viral DNA, also known as spacer. These spacers serves as a genetic memory of a previous viral infections and allows bacteria to recognize and defend against the same virus in future. Bacterial DNA that appears between each spacer is known as repeats, and both these spacer and repeats makes the CRISPR array. So CRISPR defense mechanism protects bacteria from repeated viral attacks through three main stages. This stage is termed as the adaptation or spacer acquisition, where viral DNA fragments are inserted into CRISPR array. The next step is the crRNA synthesis. Here, CRISPR array is transcribed into a long precursor RNA molecule, also known as a pre-CRISPR RNA or pre-crRNA, which is then processed into smaller fragments called as CRISPR RNA or crRNAs, each containing one unique spacer sequence with a repeat. The third step is the target interference. crRNA guides Cas protein to identify and cleave matching viral DNA sequences during subsequent infections, effectively destroying the invader before it can replicate. Cas is a CRISPR-associated nuclease protein. During this stage, an essential RNA molecule called tracrRNA or trans-activating CRISPR RNA works alongside crRNA. TracerRNA forms a duplex with crRNA and thus creates a stable complex also known as effector complex that binds and activates the Cas9 protein. This activated complex then searches for and cleaves viral DNA sequences that match the crRNA spacer. At this point, Cas9 does not cut every DNA sequences that matches the crRNA. It must first locate a short sequence which is called as PAM or protospacer adjacent motif on the viral DNA. The PAM sequence is a short 2 to 5 base pair length conserved DNA sequence downstream to the cut site, and its size varies depending on the bacterial species. Once the effector complex finds a viral DNA sequence with a correct PAM and a complementary base pairing to the crRNA spacer, Cas9 becomes fully activated. It is then it uses its nuclease's ability to cut the DNA strand, since it has two domains. So there will be two cuts at the different sites. This destroys the viral genome, thus no more new viral particles are formed. So all this occurs in three steps. One, the adaptation or spacer acquisition. Two, the crRNA synthesis. And third one is the target interference. This idea of bacterial immune mechanism was used in lab for gene editing by scientists. For gene editing, crRNA and tracrRNA molecules are combined into a single-stranded that folds back on itself and is known as single guide RNA or sgRNA. This is synthesized in the lab. When sgRNA and Cas9 enter the cell, they quickly form an active complex. This complex scans the genome for PAM sequences and matching DNA, then makes the cut. Once Cas9 enzymes cut the DNA strand, the cell starts one of the two natural repair mechanisms, which is non-homologous end joining (NHEJ) and homology-directed repair (HDR). Non-homology end joining facilitates the break directly by ligating the broken ends without any template through an enzymatic process. It is referred as non-homologous because the broken ends of the DNA are ligated with a minimal reference to the DNA sequence. It is an efficient repair mechanism that is most active in cells, but it is more prone to error mechanism that may result in small random insertions or deletions at the cleavage sites. This leads to the generation of frameshift mutation or premature stop codon. Therefore, this repair mechanism is best used when scientists want to knock out a gene of interest, because this mechanism consistently generates a small 1 to 10 base pair indel errors. These small errors can disrupt gene functions, which make it ideal for gene knockout studies. HDR, however, is highly precise and requires a homologous DNA template. In CRISPR gene editing, HDR requires a donor template containing a sequence of interest. HDR executes the precise gene insertions or replacement by adding a donor DNA template with a sequence homology at the predicted site. HDR, however, has lower efficiency compared to the non-homology end joining, as it only occurs in a certain phases of cells, the S and G2 phase of cell cycle, where homologous DNA is naturally available. Cas9 has the widest range of applications in the clinical field. It can potentially cure many genetic disorders, including blood diseases, cancers, and neurodegenerative conditions. Although CRISPR could also be used to edit human embryos or germline editing, this is still considered illegal in many countries due to ethical concerns. Beyond medicine, CRISPR-Cas9 is also transforming agriculture. It can create crops that resist disease, tolerate environmental stress, or offer better nutrition. Examples include high GABA tomatoes sold in Japan and low asparagine wheat tested in UK to reduce carcinogenic byproducts in bread. CRISPR-Cas9 has also environmental and industrial uses. It can engineer yeast to produce biofuels more efficiently or convert sugars into plastics without using petroleum.